Hello, my Bio 30 class, and I would like to introduce you guys to the first section of Unit C. And Unit C is just about genetics, molecular biology, and cell division, which is what we're going to address today. So what I want to start with is, so everyone can maybe see, I'll step over here. I want the objective today is I can explain the events of the cell cycle. And this is really, really important. This is kind of basic information. Previous years, you might have already learned about the difference between plant and animal cells, specific organelles. This is what we want to do today is go over the cell cycle or the life cycle of a cell and the specific stages that take place. So of course, our key terms today will be the cell cycle, mitosis, interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Those are a lot of crazy words, but I'll make them nice and simple for you guys. And the nice thing is, Google is a wonderful friend if you guys struggle with any of these terms. But first, I want to start with the big picture. So I wanted to do the breakdown, because primarily the cell, you think of all these stages, you kind of think they might all be equal, but this is not true. Some stages take way longer than others. Some are a very, very, very small percentage of time. So what I want to say, so here is the big section of interphase. I'll go into more detail, but it consists of 3 major phases. Then, once out of interphase, it goes into the meiotic phases. The meiotic phases are what I kind of led to in our key terms, which will be, it'll be prophase and all those other phases, which will go in order just to help you guys with the flow. It is a nice cycle. And then as well as in the end, it'll result in the division of the cells. And actually another fun thing too is there's also a point when the cell ceases to multiply. So when the cell actually dies, and it's the life cycle of the cell. So first we have interphase. And interphase is where the cell spends most of its life. It obtains nutrients, it prepares for cell division. It just performs the normal functions of an everyday cell. And like I said before, there are 3 stages. The growth phase is 1, so it's G1. It's the first phase, and it does a lot of the things I just mentioned. It obtains nutrients, performs normal everyday functions. Then there's the synthesis phase, and this is where it synthesizes or creates replication of itself. And then the last phase of interphase is the second growth phase. And this is, it's like performing those last little bits. It's still growing, it's still maturing, but it might be preparing for cell division. So for your guys' notes, I have just addressed each one specifically just so it's easier to follow. As well for my people who like visuals, I have a nice diagram. So again, so growth and normal metabolic roles, DNA replication for synthesis, and growth and preparation for mitosis. Do you guys have any questions? Is it pretty clear? Sometimes the difference between G1 and G2 might not be fully clear. Can you guys tell the difference between the 2? Yeah? Can someone give me one of the differences? It's a pre-something. It's the next phase. G2 might be. Well, if you guys don't understand, it's totally cool. This is really fresh new material. But the major difference is this is prepared after the DNA has been replicated in the synthesis phase. It's preparing for, it's a pre-mitotic or pre-mitosis phase. So it's getting that gearing up to start dividing. And so when it's ready, after the G2 phase, we're hitting the event of mitosis. And I wanted to give you guys a big picture breakdown, all of it together. So we'll start with the prophase. I'll explain that first. Then we'll go to metaphase, anaphase, and telophase. And eventually in the end, it'll be cytokinesis is the very end where they fully start separating. So next, we want to start with prophase. Prophase is nice and simple, so it's right after the G2 phase. It's immediately after. This is when the chromatin, they start to coil and condense. They want to get super tight, and they form into the chromosomes. And the chromosomes consist of 2 chromatids. And these 2 chromatids are called sister chromatids. So it's kind of nice, you think of a pair, you think, oh, we're sisters, we match, we're identical. As well as this, and we want to make sure that the nucleolus is the inside material, and the nucleus membrane, it starts to disintegrate. It's going to want to start separating from each other, but at this stage, we're just trying to coil in nice and close together so they're easier. So, because if you think all this material in the nucleus is kind of spread out, when it's condensing, it's easier, because if you think of something small put together, it's kind of easier to pull apart than say something that's flailing all over the place, taking up a great amount of space, right? So you want to remember that it's condensing, it's getting smaller. And then the centromeres, which in this diagram, they're right here, they're going to start separating, and they're going to go to opposite poles because you want to create space to separate. So one is going to one pole of the cell, the other is going to the other pole of the cell. And then as well, and so, excuse me, and like I said before, the nuclear membrane is now beginning to break down because we're going to want to start that separation. So our next phase is metaphase, and this one's nice and easy. In this diagram, it explains it really well for you guys. What we're wanting is, see, now the chromosomes, and so the two chromatids, or the sister chromatids, are paired together, and they line up nicely kind of along. It's called the metaphase plate, which is a really nice definition because you think metaphase, metaphase plate. It's a nice line. And these strings, those are called spindle fibers. And they're going to connect. So the one pole, this again is the centromere, it's going to connect to this chromatid. And the pole up, the centromere up there is going to connect to this chromatid. And so now, and I wanted to create an analogy between metaphase and anaphase. Do you guys, like Thanksgiving dinner, do you guys ever have the tradition of pulling the wishbone apart? Well, I like to make the analogy that you're with your brother or your sister or your cousin, and your arms come out like the spindle fibers, and the wishbone is like the chromatids or the chromosome, and you're going to pull it apart, but unlike the wishbone, sadly, it'll be nice and even, so maybe you can both make a wish. But they're going to be two identical pieces being separated apart. Just so maybe it kind of gives you a picture. Do you guys agree with that picture? Can you think of maybe any other ideas to help remember that you're pulling apart the chromatids? No, is it a pretty good analogy? Do you guys like that comparison?
Yeah.
Is it going to help you for remembering? I like, and while you're thinking of this, feel free to write it down. If you think of an analogy, or if you want to draw these pictures, I find they're really helpful for prompts during tests. So when you want to remember specific material, maybe remember the analogy, maybe remember, or even draw the pictures. And maybe if it would help you, draw the identical chromatids one color and then different. Because this is one chromosome. And these are, so one chromatid and another chromatid. Together, they're the sister chromatid. And then this one's a different chromosome. So this specific cell has 4 chromosomes. And so they're separating the genetic material. Is that clear for everyone? Perfect? Okay, perfect. We can keep going. So just in anaphase, all you're doing is you're separating to opposite poles. The spindle fibrils are pulling those chromatids. And then our next phase is in telophase. So in telophase and cytokinesis, I'm going to tell you this now, they're actually happening simultaneously, but they are 2 different events. So I want you guys to put that in your notes, maybe highlight it. That is a really important thing to know. So again, so the nucleus and nucleolus begin to regenerate. We'll see here that they're starting to get that cell division, which that is called cytokinesis. That'll be on the next slide for your guys' reference. So we're starting to divide. A nucleolus and nucleus are forming on this side and on this side. They're kind of a mirror of each other. And they're going to have the same amount of chromosomes. So there'll be 4 on this side and 4 on this side. It's starting to cleave. And this one I like to point out too is, so now remember we started at the beginning where it duplicated? Now there's no longer duplication. So again, for maybe in your notes, it's not in these notes, so you might want to write it down, is in the beginning, once the DNA has been replicated, it's called 2N. There's 2 amounts of the chromosomes that you need, or chromatids that you need. And then when they pull apart, it goes back to a regular 1N. So you're back to your normal amount of DNA that you'd have to for the rest of your life. So you can go on